I would like to invite Dr. Jeroen de Ridder. Jeroen is an associate professor at the Center for Molecular Medicine, Utrecht University. The floor is yours.
Yes, thanks very much for the invitation. It's a pleasure to be here. So today I'll be talking a little bit about how we leverage nanopore sequencing, one of the other sequencing technologies that was not mentioned today, I think, still, and how we use that for monitoring cell-free DNA. So I'm not sure how many of you are familiar with nanopore sequencing. Frederike Koller from Nanopore is actually in the room, so any questions can be also directed to her. And Is Nijman, who was also mentioned before, is running the sequencing center at the UMC Utrecht, and that's where all these experiments have been done. So if you, and by the way, USeq is one of the technology centers within the X-omics consortium, so you should definitely get in touch with Is or with me if you want to run nanopore sequencing yourself. So, in a nutshell, nanopore sequencing is a slightly different way of sequencing DNA, because the DNA is pulled through a protein nanopore, and then the ion flow is measured through the pore, and that gives rise to this current signal, which can then, using deep learning technologies, be converted to the base sequence that was pulled through the pore. Now, and this gives rise to very long molecules, and what's really nice is that you can sequence DNA in real time. And it's, of course, a very small device that fits inside your pocket. Now, we wanted to leverage this sequencing technology to address two very important challenges in cancer diagnostics. On the one hand, that's monitoring treatment response, to make decisions or help clinicians make decisions on dose escalation, de-escalation, maybe switching of treatments, or to determine if the patient is disease-free. And then on the other hand is recurrence monitoring, right, where unfortunately many patients experience recurrent disease even after seemingly successful treatment. So you want to know these recurrences as soon as they occur. Now, this would require very frequent assessment of the mutational burden or the tumor burden in the patients. And unfortunately, that is not really supported by the classical diagnostic modalities such as MRI scans or needle biopsies. Right? So that's why the field is now rapidly moving to liquid biopsies, which would really support this very frequent assessment of the presence of a tumor. Now, we focus on cell-free DNA, which is the result of cells dying due to apoptosis or necrosis, and in this process they shed their DNA into the bloodstream. Now, tumor cells, they also die, but tumor cells contain mutations in their genomes. So a very tiny fraction of the molecules that float around in the bloodstream actually carries mutations, and those mutations we want to find. And that essentially is a little bit of a game of Where's Wally? And because these molecules are very small, they're 150 base pairs in length, but only a very tiny fraction of them actually carries a specific mutation. You have to imagine that in a single vial of blood, there will be maybe 10 to maybe a couple thousand, if we're stretching it, of molecules that carry a specific mutation. So what we need is very sensitive detection methods to identify these mutations and distinguish them, for instance, from sequencing errors. Right? And preferably, we want this technology also to be cheap and fast and point of care, etc. Now, these nanopore sequencers, they have a relatively decent error rate these days, but it still is not compatible with this very sensitive threshold that we need to reach. And that's why we developed Cyclonic seq, which is a technology that we developed in a lab. It consists of a backbone sequence. This doesn't work, right? It does? Okay, anyway, I'll mime it. Yeah, so we developed a backbone sequence that allows us to capture target-enriched cell-free DNA molecules. So they're very small molecules, so we need to very effectively capture them into circles. And once we have these circular molecules, we can use rolling circle amplification to make copies of the same original template. And this gives rise to these very long concatenameric molecules that contain many copies of our original molecule, and these long molecules are, of course, very well compatible with the nanopore sequencing. So we sequence them on a nanopore, and then bioinformatically we cut out the inserts, which are the copies of the molecules that we're interested in, and build a consensus. And in this consensus, we effectively can average out the errors, the remaining sequencing errors that are there, and we're left with very sensitive detection of true mutations. Yeah, we can indeed do this very sensitively. We can go as low as 0.02% variant allele frequency. It's very fast, so we have 2 days to result, and that is partially due to the fact that we can already start the analysis while the machine is still sequencing. So the data processing and the sequencing is combined. And it's very flexible because we can enrich for any locus that we would like, whole genes or a panel of genes. That is all possible at the first step here. This is just an overview of some results. So on the top you see the mapping of the insert and the backbone, and that is a good read. That's what we would like to see, really this alternating pattern of the insert and the backbone. And not all reads are so nice. Sometimes we see insert only or backbone only reads, which we need to filter out. But on the right there you see a plot that shows you that the vast majority of the reads contains really this 50-50 insert backbone ratio, which is exactly what we would need. So those are the ones that are selected then for consensus calling. And that's the result that you see at the bottom, where you see the accuracy of these consensus reads as a function of the number of repeats, and you see this exponential increase in the accuracy, leveling off after around 10 repeats at around 0.999 or something accuracy, so the 1 minus single nucleotide false positive rate. But bear in mind that this is single molecule accuracy, right? So if we catch a molecule in our read, this is the accuracy that you'll get, because we get basically the copies for free due to the rolling circle amplification. So we did some dilution experiments ranging from 0% to 1%, and you see that by and large, if we apply this technology, you find approximately the same ratios back. And what's maybe most importantly, that in the 0%, the wild type case, we don't see any of the mutations anymore. So we designed a multiplex PCR strategy to amplify all the exons in P53. So that's what you see over there. We can adequately cover all these exons, and we're actually currently also working on PCR-free methods to do this enrichment, because we have seen that also already the PCR induces a few errors that we would like to get rid of. Now, based on this, we decided let's try this out in patients, and the first use case that we tried to tackle was head and neck cancer. It's quite a prevalent cancer with 600,000 new cases each year. But what's really the problem in this disease is that about 50% of the patients experience recurrent disease even after seemingly successful treatment. And these recurrences, they need to be detected as soon as possible to restart the treatment and have any chance of survival for these patients. Now, unfortunately, especially for the head and neck region, these are very difficult to spot using the standard of care, which is MRI imaging, and that has to do with the fact that there is salvage surgery or radiation going on due to the treatment in this head and neck region, and that makes these tumors very, very difficult to spot. So there's a real opportunity here for liquid biopsies, and this is in particular also the case because head and neck cancers are characterized by mutations in P53. About 90% of the patients carry mutations in this gene, at least if they're HPV negative. So that's why we set up this clinical proof of concept study funded by the Oncode Institute, where we aim to include patients that are undergoing daily doses of radiation, some intermittent doses of chemotherapy, and weekly blood draws combined with MRI images. And then these blood draws are subjected to the Cyclonic that you see over here, and a ddPCR assay, which is the standard of care to spot mutations at this low variant allele frequencies. Yeah, so this is the result for the first patient. It's a stage 2 oropharynx tumor, has a mutation in P53, and I'm clearly not an expert on interpreting these images, but even I can see from if you look at these running images that in the first few weeks after the treatment there's almost no difference in these scans. Yeah, so there's no change. It's 3 months into the treatment that you start to appreciate a response to the treatment. And it is after 9 months that it appears that there is a recurrent tumor visible in these scans. Now, if you look at the Cyclonic-seq results, that's what you see here. They're already, so this is in weeks. After the first week, you see the levels go up a little bit, presumably as a result of the treatment. Then it comes down. But instead of going down further, it actually comes up again slightly, indicating that there is detectable cell-free tumor-derived molecules in this patient's blood, which is really not observed from these MRI scans. Only 9 months into the treatment you can appreciate this from the scans. And also if you compare to the ddPCR, it actually failed at detecting this as well after 5 weeks. So this is a second patient, and the reason I want to show you is also to show you how quickly we can detect this. This is a stage 4 hypopharynx tumor. Patient with a mutation again in P53, and here you see the sequencing result as a function of the sequencing time. So it's a real-time accumulation of this mutation evidence, and you see that already after an hour or 3 of sequencing, we have ample evidence to suggest that there is mutant molecules in this patient's blood, and we can actually stop sequencing at that point and give a result back to the clinic. So we did this for a couple more patients. The clinical trial is still ongoing, but these are the first 6 patients that we've done. And you also see that by and large, the ctDNA levels correlate with patient outcome. So for instance, these patients over here, you see that after 1 year, they're still recurrence-free, and that actually corresponds with CT levels that are undetectable by our technology after 5 weeks. And you see for 2 patients, it's impossible to determine that because they died from other causes. And this patient A actually experienced a recurrent tumor, and that is also corroborated by the ctDNA result, as I showed you before. Now, based on already these first few patients, we said, okay, this is really promising, so what can we do to really make sure that this actually reaches the patient? And that's why, together with my co-founders, Wiegert Kloosterman and Alessio Marcozzi, said, okay, let's spin this out into a spin-off company. Yeah, that's currently ongoing, so that's what we did. And together also with Oxford Nanopore Technologies, we're in the process of really making sure that this enters into clinical practice. And it's a really nice collaboration between the academia, which is mostly my lab on the one hand, on the right here, and the people that are now fully employed by the company, which is called Cyclonic. So that's the first part. Now I would like to venture into going genome-wide. So we did this targeted, focusing at specific genes, specific amplicons, but there's no real need to do that only for specific genes. We can essentially try to capture whatever small molecules are floating around, capture them into these circles, and repeat our rolling circle amplification trick to get a very accurate estimate of what's in there and build consensus sequences. And in that way, you can really get a multimodal view of what's in the blood. In terms of cell-free DNA, we can, of course, capture the somatic mutations, as I've showed you before, but then genome-wide. We can also try to make copy number profiles because we really get genome-wide coverage. We can also try to look into the fragment length, which has been shown is really different for tumor-derived cell-free DNA compared to cell-free DNA derived from healthy cells as a result of nucleosome positioning and transcription factor binding. And we can also look at methylation, but that is still on the wish list, so I don't have data on that yet. So we first applied this to ascites samples. So this is a result of a buildup of fluid in the abdomen of ovarian cancer patients. And the reason we use this as a first use case is because there's a lot of it, so we can really do repetitive testing on the same sample. And typically the tumor fraction is quite large. So I'll show you the first results on a few ascites samples, where we indeed employ this genome-wide Cyclonic-seq technology, building consensuses. So this is just to show that we can actually detect mutations. Here's a couple of mutations. The top tracks are the Illumina sequencing result, where you can clearly see that there is a variant detected. And then at the bottom row, it's hard to show you, but maybe yeah, there you go. That's a single cyclomic-seq read. And you can see that in a single read, we actually capture the same variant. And we get, of course, the same coverage, actually deeper coverage than with the Illumina. This is about 10x Illumina. We get deeper coverage, but it's kind of intrinsic in our method because we get these copies as a result of the rolling circle amplification. And you see that also these Illumina reads are not super clean. There's actually errors for which you really need the read depth. On the right, you see that we actually can capture through somatic variants. We cover about 4,000 of these somatic variants out of the 13,000. But what's maybe more interesting is to look at this VAF profile, which really follows, if you look in the blood, it really follows the variant allele frequency profile that you observe in the solid tumor, indicating that we are really truly capturing somatic variants from the tumor. Here is some genome-wide copy number profiles. We're showing you that we can really get genome-wide coverage, and we can accurately recapitulate the copy number profile as we observed it in the solid tumor. At the top, you see the result of 1 minion run, where we get about 3.5% coverage of the genome, which is actually sufficient to make these kinds of plots. And of course, the results improve a little bit if you throw in a little bit more beefy sequencing. And at the bottom, we use the Promethean machine to get more sequencing reads. And then you indeed get a slightly improved copy number profile. Depending on how you want to use it, of course, you would either choose a minion run or a Promethean run. And then the fragment length distributions. So we know that the tumor-derived cell-free DNA molecules are a little bit shorter. And that's actually also what you see here on the left. In orange is an ovarian cancer sample compared to 4 healthy controls. And you see that indeed there's this shift of the peak, a little bit to the left, a little bit to the shorter end. But what's now interesting is that our read length is, of course, not limited to only 250 bases or so, but we can really go up to 1 kb. And there you also appreciate this shift to the left, to the shorter end, which is, however, much bigger. Yeah? So that is really showing that it is important to also look at the slightly longer fragments that are floating around in the blood. So having said that, to summarize, so Cyclonic-seq really enables accurate detection of cell-free tumor DNA in the blood, in liquid biopsies. And we can also do this genome-wide, and that really allows multimodal integration of different analytes or different types of measurements that you can do on the cell-free DNA in a single assay. So with that, I would like to thank the people in the lab, the people at Cyclonic, but also certainly the people at Nanopore with whom we're really taking the next steps. So thanks a lot.
Thank you for sharing this very interesting work with us. We have time for one question.
So I was actually appreciate the talk, but I was thinking a technological question, because you're using nanopore sequencing with a biological nanopore, but there are new generation technologies like solid-state nanopore techniques. So do you think that they will at certain moment supplement or replace the technology that you're using? What's in the future?
Yeah, the person behind you is maybe the best person. She's the representative from Nanopore, and she will tell you that the protein nanopores are fantastic. But at the same time, I've been around conferences, and I know that maybe the solid-state nanopores are around the corner, which would potentially really increase majorly the throughput that we could see from these devices. So yeah, we'll keep a keen eye out on these developments, because they could certainly bring this to the next level, right? Yeah.
Thank you.
Thank you again. And here is a little chocolate bar.
Thank you very much.